Okay, Assalamualaikum warahmatullahi wabarakatuh. We'll be discussing the plant pathogen identification using molecular identification. Now, first, we will learn about the terms of diagnosis, identification, and also detection. These three terms that are essential in plant disease protection, and you shouldn't be confused with one another. So the diagnosis and also identification and detection, yeah, all three, these terms are process. Yeah. And for the example, the diagnosis was intended on the disease. Yeah. So the target is the disease. What the disease? Yeah. The occur in this plant. And for the identification, the target is a pathogen. So we identify what the pathogen, and the detection was intended on the existence. Is there any pathogen or not? Yeah. Is there any specific pathogen or not? Yeah. So when we want to know the disease, now we can go to the diagnosis. If we want to know the species or the genus of the pathogen, we can do the identification. Or if we can like curious, is there any pathogen? Is there any virus in that plant? We can do detection process. Okay, so before we diving deeper, yeah, let's refresh some basic knowledge. You may recall the term of genome. So genome is the entire set, entire genetic material of an organism. So it's much like a computer program or a smartphone application that built with code. All biological functions within our body are also written within our genome, yeah, in code. So this genome includes sequences of DNA or RNA. Yeah. So the DNA or RNA containing the genetic instruction, yeah, encoded in the language of four bases: thymine, adenine, guanine, and also cytosine. Yeah, A, T, G, C. Yeah, they're always pairing A with T, C with G. Yeah. Okay, so the DNA is a various nucleic acids that are usually the molecular basis of the heredity and constructed of a double helix. Yeah. You can see in the picture, the DNA is a double helix that held together by hydrogen bonds, yeah, between purine and pyrimidine base, yeah, ATGC. Okay. So this bases, yeah, ATGC are arranged in sequences that form genes, the unit of inheritance. Yeah, so we know the gene here, the unit of inheritance that may be expressed as a protein essential. Yeah, so gene is a specific sequence of nucleotides in DNA or RNA that is located usually on a chromosome. Yeah, so sometimes we call rDNA. Yeah, because it's located in ribosomal. Okay, so and the gene also, especially a protein, yeah, controlling the function of the other genetic material. Okay, and nucleotide. So nucleotide, several compounds that consist of ribose or deoxyribose surkin joined to a purine or pyrimidine base and to a phosphate group. And that are the basic structural units of nucleic acid. Yeah. So the basic one is nucleotide. Yeah, the whole informatic is a genome. Yeah. Genome containing DNA, and part of DNA we can say as a DNA sequence or DNA fragment. And on the DNA fragment, we can see the gene that containing nucleotide. Okay. Now we move to the identification. Let's look at how we differentiate species. So identification relies on specific characteristic or traits, yeah, traits that unique to each species. What makes different, yeah, between these insects are about the unique characteristic, specific characteristic. So for to distinguish one insect from another, yeah, or one fungi to another, we need to identify features that unique from other species. Okay. Okay, so for the example, yeah, we have some species here from A, B, C, D, E up to T. Yeah, and then there are a lot of characteristics from character 1 up to character 9. Okay, so we can see here in the E species and also the I species, what characters that we can that can be basis of identification from character from species E and from species I. Yeah. For the first characteristic, yeah, we can see here the color of the E is orange, and also uh, in the I species, the character of the character, first character is also orange. Yeah. So we cannot identify by this characteristic. Yeah. So we can find other characteristic that different between species E and species I. For in the example, from the fifth characteristic, yeah, we can see here the, oh no, it's same, yeah. From the fifth species, it's same, yeah, the blue colors. Yeah, we can go to other what different. This one from the 8 characters, yeah. Oh, sorry, sorry, it's a mistake. So, from the fifth characters, yeah, we got this blue and we got this orange, yeah. And also for the sixth character, this white and this blue, yeah, yeah. So we can identify, is it characteristic, is it species E or is it species I by using characteristics of 5 and 6, yeah. For the example, the characteristic of Five is about the nose, yeah, and the characters of stick is about the mouth, yeah. So we can different the characteristic of species E and I by observe the nose and the mouth, yeah, for the example. Okay, so go back to the molecular terms. Yeah. So we have known that the traits are encoded in nucleotide sequences. Yeah. On the sequences of nucleotide. For the molecular identification, we utilize nucleotide sequences that are consistent and unique to each species. Yeah. So there are nucleotide sequences that are consistent and unique to each species. Yeah. So there are internal transcribed species. Yeah, we can see ITS and also other regions in the genome that can serve as reliable markers, yeah, for the marker for species identification. Because some of the nucleotide sequences are the same between one species to other species, but there is a nucleotide sequences that different between species, and it is used for the marker for species identification. Uh, the region that consists of specific nucleotide sequences for the example are internal transcript spacer or ITS. Yeah. Okay, you can see here, yeah, the nucleotide sequences from different species. Yeah. So there are two different species, E. trifoli and E. diffusa. Yeah. So in the E. trifoli and E. diffusa, in the first nucleotide sequence, it's same, right? Yeah, it is same. Yeah. For the last one, yeah, the E. trifolis are TCG and the CTG is for diffusa until the end. So the difference of this base, yeah, the nucleotide sequence, yeah, will help us to identify the species. Yeah, for the example, yeah, we have a isolate A, B, C, D, and we have a known what is the species of the species isolate A, what's the species, B, C, and D also. We don't know the species, but we have nucleotide sequences here for the isolate A, G, 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 T, T, yeah, and so on. Isolate B, A, A, T, T, C, C. Nah. And here we have a data. Yeah, we have a database. Yeah, and knowing the nucleotide sequences in a specific region. Yeah, for the example, species 1, the nucleotide sequences are AAA, TTT, GGG. Yeah. So that is a website that contain database of microorganism, yeah, genetic information of microorganism include nucleotide sequences. Yeah, you can go to the NCBI. Yeah, NCBI. It's a database of molecular identification of organism. Anything. Yeah, include human, animal, plant. Yeah, microorganism. Yeah, if you have a nucleotide sequence that you get from molecular identification, then you can uh, insert your nucleotide sequence that you have got to the website, and then the website will process, yeah, what species is related to your nucleotide sequence. Yeah. Okay, I will make it colorful, yeah. So it's easy to observe this uh, base, yeah. So the base isolate A's, G, 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 T, 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 C, C, A, A, and so on. And it is similar with the species 4, yeah. Is that right? G, G, G. TTT, CCAA, CCAA, TTT. Yeah. So we can conclude the isolate A is similar with the species 4. It means the the isolate A is a species 4. Okay. Uh, so yeah, for the molecular identification principle, yeah, the principle is about conserve and consistency. So they are region in our gene that consistent in the organism that consistent, not change. Yeah. Uh, Even the environmental condition or other condition, the gene always the same in each species. Uh, and the region of the nucleotide sequences that always consistent is a ribosomal DNA internal transcript spacer or ITS and also intergenic spacer or IGS. Yeah, you can see here. So we have a rRNA gene cluster here. Yeah, uh, that being a walls between the intergenic spacer. Yeah. So we got the ITS1 and also the ITS2. Okay. This is the region 18S rRNA and 28S rRNA. Okay, so the image displays the structure of our DNA repeat regions and sequences that commonly used in a molecular identification. So the rDNA gene cluster consists of repeated rDNA units. Yeah. Yeah, maybe we can see in this next slide. Yeah. This one. Yeah, so the rDNA gene cluster consists of repeated rDNA units that show and in a purple color, this one. Yeah, with non-transcribed spatial or NTS separating each unit. So each rDNA, yeah, each rDNA contains several critical components, yeah. 18S RNA, so 18S rRNA, sorry, is the part of the small ribosomal subunit, and also 5.8S RNA, sorry, located between 18S and also 28S RR genes. And we also see here 28S RNA is the part of the large ribosomal subunit. Yeah. So the ETS, yeah, external transcript spacer ETS or internal transcript spacer ITS, yeah, it's the same, yeah, are included in each rDNA repeat units. The ITS regions, yeah, ITS1 and ITS2 separate the rRNA chains that highly variable and of course making them useful markers for species level identification. So the image, yeah, this right side and the left side also includes two blocks of nucleotide sequences. Yeah, one is belong to Fusarium oxysporum and Fusarium fujikuroyi. Yeah, so likely representing regions within the rDNA cluster, yeah, such as part of the ITS or 18S or 20S regions. These sequences can be used for comparative analysis in molecular studies, yeah, to identify species or to analyze the genetic diversity. Okay. Okay. And for the PCR, polymerase chain reaction, maybe I will explain in a separate session, yeah, the next week. So thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.